Before we start lab this semester, a little practice with pencil and paper will get you prepared for what we will do in our lab activities. Practicing streak techniques is a very effective way of preparing for lab. We have 4 very different streak techniques that we use in microbiology. There are other types of techniques, but these are the ones that we will use this semester. One is a urine streak. The second one would be streaking for a confluent lawn of bacteria. The third is the quadrant streak for isolated colony-forming units, also called colonies. And the last and fourth technique that we will do this semester is what I call the pie section streak technique. Now, the general purpose medium that we will use routinely all semester is called trypticase or tryptic soy agar, TSA. That's the gel that looks a little bit tan or beige in this photo. The TSA agar is in a plastic container that's called a petri dish, which we also call plates. So petri dishes are the same thing as plates, and the agar is the bacteria food that's put into the petri dish. Notice that TSA agar is a general purpose medium that grows non-fastidious and semi-fastidious bacteria. It does not selectively grow pathogenic bacteria. It's just a general, all-purpose medium that most bacteria will grow on, that grow in and on the human body, that we routinely recover for clinical evaluation and samples. Paper practice first, I've found, has been very helpful for students and saves materials, so we can be very cost-effective. On a piece of paper, draw a circle that's roughly 100 millimeters in diameter. This is the size of the petri dishes that we will use this semester. With a pencil or pen, or even a marker, practice drawing the following example techniques that are illustrated on the additional slides. For urine, confluent lawn, quadrant for isolated colonies, and the pie sections. Bring this paper and all these paper copies of your practice activities with you to lab the first day of the semester. Lab activities. This is just an example. It's just a basic circle. You could either use the version that's on the PowerPoint that accompanies this same information on our course online site, or just draw what you have on a piece of paper. For the urine culture streak technique, we're actually going to perform this technique in the laboratory with fake or artificial liquid cultures or liquid preparations. And to do a streak technique, we're going to take a loop full of bacteria, streak it right down the center of an agar plate. That was called the primary inoculum. And then at 90 degrees, cross streak with the same loop, not doing anything else with it. After we've applied that primary inoculum down the center, do at least 12 cross streaks at right angles to that primary streak. And that's it. So the urine streak technique, I think, is probably the simplest and easiest one for students to learn. There's a link that's also found, again, in the PowerPoint with the same information on the course online website, or you can go to that link as well for more information and more examples of urine streak techniques. Hopefully, when we come back for our second lab day, after we've streaked our urine cultures, our urine streak plates will look a good bit like the photo on this slide. It's a semi-quantitative technique. Some will tell you it's quantitative, but we found that usually the number of bacteria that are sampled are so numerous, we can't get a really accurate count. So we have to estimate the number of bacterial cells per milliliter of urine, but we can get it pretty close to what's actually there. The second technique that you need to practice before you come and actually do it on a real agar plate is the confluent lawn streak. For this technique, we want to cover the entire surface of the agar plate with our bacteria. We will use a swab. We'll learn more about the tools when you get into the microscope lab, microbiology labs, but we have cotton swabs that look like the cotton swabs you purchase at the drugstore or at the grocery store or any other store. And we also have metal loops. It's just as it says. It's a little wire with a little loop on the end. Different techniques require different tools. And for the confluent lawn streak, we're going to use a sterile swab. We're going to collect a little bit of sample of bacteria with our swab, and then for the first pass, we cover the entire surface of the agar plate in one direction, going from side to side of the petri dish. Cover the entire surface and keep your streaks really close together, even closer than what I've illustrated on the first circle with the black marker. Then you're going to turn your agar plate, or in this illustration, you're just going to cross streak that first set of streaks in black. Notice that I've cross streaked over it in red. And you want to, again, keep your streaks really close together, closer than what I've illustrated. And you want to go all the way to the edge of your circle, which is our petri dish. You're going to turn your agar plate another 45 degrees, or a sort of a half turn, a little quarter turn, and you're going to cross streak again. So we've got the black streaks underneath going in one direction, the red streaks going perpendicular to those, and now we have these little green ones sort of obliquely across everything. Again, you want to keep your streaks really close together, closer than what I've illustrated on this drawing, so that we have a nice, complete, what we call confluent lawn, completely covering the entire surface of the agar plate. So we've spread the bacteria 3 different directions, so that when we come back the next day, we will see that entire surface of the agar plate has bacteria growing everywhere. This is a fairly common technique that's used for many different applications in microbiology. The third technique you need to practice with pencil and paper is the quadrant streak for isolated colony forming units, also called CFUs, which in lab we call colonies. Same thing. Just one form or one phrase, colony forming unit, is much more formal than sort of the nickname colonies that we routinely use in lab. Quadrant streak, as the name implies, has 4 sections. They're not all the same size, and we are going to use a metal bacteriological loop in lab to do the real streak. On pencil and paper, or pen and paper, you're just going to practice doing the streak technique and keeping your streaks in nice, precise sections. So, in the real lab, you're going to take a metal loop, you're going to sterilize it, and you're going to take a little bit of bacteria from a source that we're going to give you and apply it to this first little section. We call it the primary quadrant. And notice nice sharp Z's for the streaks. Keep them really close together. Don't scribble back and forth. You start in one spot and you just pull those out so that you have nice separate Z streaks for your primary quadrant. Now, in lab, we're going to do a technique that we call sterilizing our loop. So we want to use a nice, clean instrument, but we're just using pen and paper right now. So to create your second quadrant, notice we're going to overlap our second section over our first, again with nice sharp Z's back and forth. And in the real laboratory, you're not going to go back and resample the original source of bacteria that you used to make your primary quadrant. The quadrant streak technique is a dilution technique. We're going to simply apply a little bacteria in the primary quadrant, and then we're just simply spreading it out. We don't go back and sample again. And students often get very confused about this because it's hard to believe that there are so many bacterial cells in that tiny little smudge that you put on the primary quadrant. How could there be enough? But trust me, there are plenty of cells. So practice doing this nice Z technique of overlap between the second and the first quadrants. For the third quadrant, you're pretty much going to do the same technique you did for the second. Do nice sharp Z's. Notice that all of them go right to the edge of the petri dish or our circle. And again, you're diluting out, you're spreading out the bacteria. Imagine that you're separating the cells with your pen and pencil, and you'll do the same thing for real with your bacteriological loop. Now going from right to left, our last section or quadrant, we're going to overlap with nice sharp Z's between our fourth and our third, but notice there's something different about this last section. We're going to have some separate, well-distinct Z's that are covering what remains of the clear space on our little drawing of our petri dish, and we're going to do the same thing in the real lab activity with our agar plate. We want to have these nice Z separated streaks on what remains of the clean surface of the agar so that the bacterial cells can form nice separate distinct colonies. That's why we call them isolated colonies. Once they're isolated colonies, we can identify single species. But this is simply to do paper and pencil practice of the streak technique so that you'll have more proficiency right away when you do the real thing. The last streak technique is simply a type of general technique that we call the pie sections to give us an idea if we're using different kinds of media, if the bacteria can grow or if it won't grow, and if it does grow, is there any change of color in the bacteria? Is there any change in color in media? So there are lots of reasons we do these little pie sections. And it's a very economical way for us to do a lot of tests on one agar plate. So again, draw your circle, and then you can divide it up into however many pie sections, at least 4. So this one illustrates 5 pie wedges, but you could do 4, you could do 8, you could do 6. Just so as long as you have at least 4 or so pie sections. And notice that these are nice little streaks. We try to keep them, again, we use the Z-like motion and try and keep them separated. So we have a little bit of an easy look, an easy image to view when we come back and actually do the real thing in our exercise in lab. So again, this is simply for practice with pencil, paper, or marker and paper, and then once you've done these little practice papers, please do bring them with you to lab for our first lab day of the semester.